Good morning, everybody. Welcome to the Institute of Physiology of the Czech Academy of Sciences for this event, which is called Day of Physiology and Medicine for undergraduate students. This day we organize for students because we would like to offer you the possibilities for bachelor, master science, and PhD thesis. And first, my name is Olga Zimmermanova, and I am the main organizer of this event. And for the introduction to the research, what we do here, I would like to ask the director of the institute, Jan Kopecký, that will tell you a few words.
Okay, thank you, Olga. Thank you for coming here. You are apparently a fraction of the most motivated students, and we are very glad that you are coming to see what's going on here. So I will give you a very short introduction, and then you will have a chance to see what's going here in practice. So this is a picture of one day at the institute, and you see how many people are approximately coming here. Okay, so this is the group of people that is working here, and as you see, we should have more of them coming. We should have about 400 employees. So we are one of the biggest institutes of the Academy of Sciences. In the Academy of Sciences, there are I think 54 institutes, and we are one of the large institutes. So you are now at the Institute of Physiology. We have 3 main areas of research. I will tell you in a minute. And there is a lot of scientists and also 80 PhD students coming here. So you might be one of those. Our buildings are located here at the campus of Biomedical Institutes in Courage. Maybe you know already the area a little bit. There are some other bigger institutes of the academy, like Institute of Microbiology, Institute of Molecular Genetics, Institute of Experimental Medicine. So we are one of those. This is a cluster of biomedical institutes of the academy. And another similar cluster of life sciences institutes is located in České Budějovice, but here we are more linked to medicine, clinical science. So you will understand when you will pass through the day. And what is very special about our institute is that it was funded by prominent professors of medicine. And this year we celebrate 70th anniversary of the institute. So really 70 years ago, on the 1st of May, this institute was funded, and there were a couple of very important and influential most all of them, as I showed you, professors of medicine, and each of them had his special interest and was actually a founder of one field here, actually. And there were people involved in muscle physiology, cardiovascular research, and what was very specific from the very beginning, that these persons were very much interested in ontogenesis, in development of the individual, what's going on from birth till the old age, because what's going on at the beginning is somehow linked to the metabolism and development later on. So these guys were the founders. And this was very important area for the whole Czech science. This was really foundation of something very new here. So we are following these 3 fields until now, and different labs are somehow involved in these 3 different fields. Metabolism, it's a quite broad description. We are, of course, focused only on some specific aspects of metabolism. So this is a general scheme how we operate. We are here experts in basic mechanisms, these basic biological mechanisms. We are studying in animal models and also in cell cultures and at the molecular level. And then to understand what's going on in human body during etiopathogenesis of diseases, we have collaborations with clinical scientists. So we are combining experiments in animals with clinical research. We have good contacts with a couple of clinical centers around here. The closest center is across the street, Institute of Clinical Experimental Medicine, but we also collaborate with scientists at other places in Czech Republic, which is very important for us, what we are doing. So our mission is to characterize basic biological mechanisms with the aim to improve prevention, diagnosis, and treatment of major non-communicable lifestyle diseases. You know approximately what I'm speaking about, right? And as I told you already, what is specific for our efforts is that we are really interested in the ontogenesis and the links between perinatal development and aging. When we talk about non-communicable lifestyle diseases, we know that everything is interlinked and obesity is very much at the beginning. You know that in the Czech Republic, but also in other countries, we have a big problem, still growing problem of obesity. Population is getting fatter and fatter. And this is not the disease obesity, but it is linked with diseases. And these links are very, very strong. And the strongest association between obesity and a disease is the correlation or association between obesity and diabetes. So if you are obese or overweight, you have a very high chance that you will get diabetes type 2 when you are getting older. And there are, of course, some other diseases also associated with obesity, but diabetes, this is the major problem because about approximately 10% of the population, and maybe even more in the Czech Republic, maybe 20% of population is suffering from diabetes. Some cases are not recognized. So this is a really very big problem. And this is not only a problem because you have diabetes, but again, diabetes is associated with some other pathological states. And there is a very strong association with cardiovascular disease. So if you are a patient with type 2 diabetes, your chances that you will have ischemic heart disease are about 3 to 4 times higher than in the person without diabetes. And what is very strange, that in the Czech Republic, the incidence of cardiovascular disease is 3-fold higher than in France, for example. So there must be some factors that we still do not know in very detailed. We know approximately what's going on, but this is a very, very, very specific situation also in the Czech Republic, that we have so big troubles with cardiovascular diseases which are linked to obesity and diabetes. And because of these cardiovascular diseases, you have a chance that each second of us will die eventually of cardiovascular disease. This is like that. More than 25% of expenses of the whole healthcare system is somehow spent because we need to treat complications of diabetes. And even worse, diabetes is linked even with the development of neurodegenerative diseases of central nervous system. You know, Alzheimer's disease is something which is really awful. If you are getting old, your chances that you will suffer from Alzheimer's disease are also very, very high. And this is correlated with diabetes. So all of these things are interlinked. And we are studying specific aspects of development of these diseases. We want to understand the mechanisms, to understand what are the targets for treatment. This is our mission. And you know that there was a COVID pandemic now, and all these situations was even exacerbated. So even worse during the COVID time. So now COVID is over, but there are still some problems that remain. So what is specific about this non-communicable lifestyle diseases? You can say each of them is different. But what is very interesting, and what I also like, that there are many aspects that are in common. And there are very important variables that are behind development of these lifestyle diseases. As you know, genetic background is very important, right? Each of us has plenty different genes, and the combination of genes is better or not so good for development of these diseases. And then very strong influence of the environment, lifestyle, right? You know well. Then these diseases are developing with aging, and some diseases are also more pronounced at depending on the sex of the individual. And for us, when we are studying the mechanisms, we use similar models. Like we need clinical cohorts, we need different models of the animal different models of the disease in animals. We use cell cultures. Then we dissect tissues from the animal. We study ex vivo system. We study things at the molecular level. And there are mechanisms that are interlinked in the development of the diseases: inflammation, energy metabolism, everything you can imagine in pathophysiology is somehow involved here, more or less. And because of this complexity, to be able to understand what's going on, you have to have possibilities to use different methods, from the whole phenotyping to molecular level. All of these approaches are more or less developed here, and this is just an illustration of the instrumentary that we have. We can study behavior at the whole in whole animals. We can characterize metabolism of the whole animals, and we can see what's going on in tissues, how much fat the animal has. And then we go deeper and deeper, and we finish at the molecular level. So during last years, we developed so-called metabolomics and proteomics, which are very powerful approaches. So from one biological sample, you can get 10,000 pieces of information. But what to do with this information? Then you have to develop bioinformatics, because otherwise you are lost. So now we are focused on developing bioinformatics. We have all these approaches here, but we have to understand what's going on. So this is approximately the state of the art. And what is very important for us to have a good animal facility. So during the last 4 years now, we reconstructed our animal house. We are at the end now. This year we will open the animal quarters, so we will be able to do experiments in animals in a very nice environment. And when you do research, you need money. And this is quite fortunate now, the situation, because we are involved in two big consortia in the country. One is focused on metabolic and cardiovascular disease, and the other one is focused on neural sciences. And thanks to this consortia, we have relatively good support, at least for 2 more years, for this year and for the next year, which may be important for you if you would like to come here, because we can support PhD students that are motivated to do nice research. And we have a lot of partners that you can collaborate with. You can collaborate with clinical scientists, but we can also collaborate with other people in science. So we need motivated PhD students if we can offer something, right? And this is just the example of the fields that are typically studied by you here, or by PhD students. And you see that our students have a very high rate of success. Most of them defend their PhD thesis, which is good for us and for you as well. So I will end here, and I will ask Olga to explain more practical aspects of your visit here. And I hope that you like the day here, and some of you maybe will get interested and will get in touch with some individual group leaders, and you are most welcome.
Thank you very much. And we will continue what we offer you for today and also generally during the year. So students and IFES are here always in cooperation with a number of Czech universities. Again, you can see here the various subjects in which you can do your study. The blue one are the most popular here. And currently we have opened the PhD call for the PhD program which will start in autumn 2024. There is an online application form at our webpage, and the deadline is 24th of March. And today you will hear the available positions, and the list of them is also on our web pages. During your PhD, as you will see and hear also from our free selected representatives of PhD students at the end of this morning or early morning part of the day, there are several benefits that we offer for PhD students that besides the main is that it's a research in neuroscience, cardiovascular physiology, and metabolism. You will learn a lot of physiological, biochemical, and molecular biology methods. And you can be employed in our institute. It depends on the budget of particular laboratory. It's besides to your scholarship from the university. And we participate regular events for PhD students like seminars, meetings, and every 2 years, PhD students meet together somewhere outside of Prague and present their results there. And in the third year of PhD study, there is an advancement report, something like checkpoint if your PhD study proceeds well. And before the final defense at the university, you have the opportunity to pass the internal PhD thesis evaluation here at the institute that can improve your PhD thesis. We have also English language courses, and in this green campus, you can find sports facilities and also on-site accommodation. And for bachelor and master science program, you will again hear the possible topics that you can follow in our institute today. But again, list of available topics are also at our web pages. And in this case, I recommend you to contact particular supervisor to write him or her an email and ask if there is this possibility to do the PhD thesis or to start the master science thesis. And here is the program of the morning session. And I would like to ask, and after there will be a coffee break, and at the end of this morning session, you will have a refreshment in the next room, and then you can visit up to 3 laboratories according to your selection. And during the coffee break and this refreshment break, you can register there on the list of papers and sign there your name, and we will know which laboratory you would like to visit. And this is the end of our introduction. I wish you to enjoy this day. And I would like to ask Jussi Palecek, head of the Laboratory of Pain Research, to present his topic in his laboratory.
Thank you. I'm glad that you came here, and I hope you will have a nice day, even though we will start with some pain. I hope you understand what we will talk about, and I hope it won't be that much painful. So what we do, we really study the mechanisms of pain in order to try to develop some new approaches and possible new drugs for pain treatment in patients. I guess that maybe you know something about pain, and you have to realize that there is acute pain, which is an important physiological signal. If something hurts, you know that something wrong is happening, so you can try to avoid it. On the other hand, you have also chronic pain syndromes, which is really a pathological state, which has really deep negative impact on the patients. And there are also different types of pain. So there is pain which we call nociceptive pain, and that's related, that's the pain that you are probably used to, that if you burn your finger or cut your finger or something, you activate nociceptors and you feel it as a painful. And we have some good drugs to treat pain, even though they are not completely perfect. On the other hand, there is also a thing which is called neuropathic pain. And neuropathic pain starts with damage to peripheral and central nervous system. So you can imagine like a nerve injury or diabetic neuropathy. You can imagine damaging by cytostatic therapy, ischemia, and other possible injuries. And what is important is that mechanisms of neuropathic pain development are different from the nociceptive pain, because the nociceptors are not really activated in the periphery, which is signaling normal pain, but the pain is developing from the pathology of the peripheral nervous system, from the nerves. And we don't really know which are the exact mechanisms, how the pain develops. And that's also the reason why we don't know how to treat it well. And there is really a high unmet need to develop new analgesic drugs, especially for these neuropathic pain states, because the patients don't have really many possibilities how to treat it. So in general, where we can stop pain, if you think about the pain pathway coming from the injury, let's say in the hand, so we have some peripheral targets, nociceptors for the nociceptive pain. Then you have spinal cord with synaptic modulation of the sensory information coming from the periphery. And then you have brain, where is the perception of pain, because pain is really a cortical mechanism. So what the nociception is really the mechanisms of activating nociceptors, peripheral targets, and pain is really becoming a mechanism in the brain. So what we do in the lab, so we have several projects which are concentrated on the neuropathic pain, especially the chemotherapy-induced and diabetic neuropathy, the role of neuroinflammation in the peripheral and central nervous system involving different chemokines, the role of TRPV1, maybe you know TRPV1 receptor transient receptor potential 1, or also called capsaicin receptors. You know about capsaicin, right? Who knows what is capsaicin? Raise your hand. Okay, perfect. Hot chili peppers. So we can induce by chili peppers pain, but these receptors which are activated by capsaicin can be also used for treatment of pain and also modulate nociception. And we are also interested in the interaction between opioid and cannabinoid receptors. And we have projects which are looking for new drugs using neurosteroids in cooperation with Institute of Organic Chemistry and Biochemistry. Methodologically, we are using our major method is electrophysiology. So we do electrophysiological patch clamp recording. Who heard about patch clamp? 1, 2, okay. So patch clamp you can see down here. It's generally a glass pipe patch which we can attach to a neuron. In this case, it's a spinal cord slice. So there are a number of neurons. You can see this pipette is attached to a single neuron in the dorsal horn. You can attach the glass pipette. You pull either a patch or you just break the membrane of the neuron, and you can record currents from the single neuron. So you can record what is the activity of the ionic channels of the currents and look how, let's say, the synaptic activities change and other things. We can also use optogenetics, optical stimulation of specific population of neurons, calcium imaging, different behavioral tests, because of course the pain is central mechanism, so we have to test behaviorally, and other biochemical methods that you probably know quite well. So as an example of what we do, here is the model that we are also interested in, the chemotherapy-induced peripheral neuropathy, which is really a serious effect of different cytostatic treatments in the patients where they are treated for cancer, but they develop neuropathic pain when they feel, burning pain in the extremities, or they can feel low touch pain, mechanical allodynia. And there is no really very good therapy for these patients. So we are studying the mechanisms, the peripheral mechanisms, and also synaptic mechanisms at the spinal cord level, where we are interested in what are the modulators of synaptic transmission, how does it change, what are the receptors and neuroinflammatory mechanisms involved in development of this pain. And we are looking, of course, for targets for possible treatment. So we have published a number of papers in this area, and we show that there is involvement of immune cells and also toll-like receptors 4, that there is activation and sensitization of TRPV1 receptors, and that there is involvement of different kinases, especially phosphoinositide-free kinase. And with that, we published our last paper in Journal of Neuroscience, and I would like to show you some of the results to give you an idea. So this part is concerning behavioral experiments. So we were testing mice in this case for the sensitivity to mechanical stimuli. So we have it's called von Frey filament, and you are approaching the hind paw of the animal with a light filament, and you measure the force by which the animal will lift the paw. So you see under normal conditions, it's about 70 grams. These were rats, I think 70 grams. But if you give these animals cytostatic treatment, like in the patients, in this case paclitaxel, you see that the sensitivity will increase. So they will become more sensitive to light touch mechanical stimuli, which is the one of the aspects of the peripheral neuropathy. And you can see that if we treated these animals with duvelisib, which is an inhibitor of phosphoinositide-free kinase delta gamma subunits, which is also used for some treatment, approved for treatment of patients, which is interesting in some hematological treatments, we prevented generally development of this mechanical allodynia, this increased mechanical sensitivity. We have shown these effects also in the patch clamp recording in spinal cord slices from those neurons. So this is a recording of activity of single neuron. You can see there is a lot of activity, which is enhanced by capsaicin application, which is activating TRPV1 capsaicin receptors in the spinal cord. So not these peripheral, but on the primary afferents presynaptically in the spinal cord of the horn. And you can see that under normal conditions, the activity to capsaicin is about this high. But after the paclitaxel treatment, it's highly elevated. So, and again, the duvelisib prevented this elevation, suggesting that it blocked the sensitization of TRPV1 receptors. And this is an example of the optogenetic optical stimulation. So these are inhibitory currents, which are evoked by activating neurons, inhibitory neurons, which have channel rhodopsin in their membrane. And when we light a light blue light on these neurons, you see for 5 milliseconds, we can activate huge inhibitory currents, which are evoked by release of the GABA and glycine in the slices. And again, we can check the changes, and it is known, and we've shown here as well, that during the peripheral neuropathy, the inhibitory currents are decreased, meaning that the inhibition is decreased. And that's also one of the mechanisms why the patients, and in our case, the animals, there could be enhanced sensitivity to pain. And we also have done some immunohistochemical staining in those root ganglion neurons. These are the primary sensory neurons, which have extension of one dendrite in the periphery, the second in the spinal cord, those horn, and we have shown changes in the expression of phosphoinositide-free kinase. So that's about just give you a hint what you can see. This is the lab. You can see we have a few PhD students. Our postdoctoral students are recently now one in Hamburg in Germany. Pavel should come back in next year. Mario just went to Texas. He's there for 1 week, enjoying sun. And we are really looking for some pre-graduate students for bachelor master science studies, probably at the Charles University, and also for some PhD students if you are interested. So if you are interested, you can come in the afternoon to our lab, and you will see some of the laboratory environment, and you can ask more detailed questions. Thank you.
The next presentation will be from the Laboratory of Neurochemistry, given by Jan Jakubík, and the head of this laboratory, and then by Helena Janičková.
So good morning, everybody. I will have very, very brief presentation of our one project, which is covered by NPO grant, which focus on mechanisms of selective modulation of individual subtype of muscarinic receptors. We are looking for bachelor, master, as well as PhD students. So muscarinic receptors are receptors mediating function of acetylcholine, and there are 5 subtypes of muscarinic receptors. And based on the localization, and subtype, they modulate various physiological functions, from vegetative functions like heart rate and intestine motility and so on, up to the higher cognitive functions like cognition, memory, and so on. So if the signaling via muscarinic receptors is somehow disturbed or changed, then these changes in muscarinic signaling is connected or associated with various diseases that involve like central nervous diseases, signaling via muscarinic receptors is somehow changed. So it is translated to various diseases that may be Parkinson's disease or Alzheimer's disease, schizophrenia, or some peripheral diseases like chronic pulmonary obstruction disease, irritated overactive bladder, dry mouth, and so on. And that's why muscarinic receptors represent some important pharmacological target. We can either block the receptors or negatively modulate. It's the left column to treat this disease, or somehow positively modulate or activate these receptors, this right column. So as you can see, if we modulate muscarinic receptors, we somehow affect a lot of things. So to treat a particular disease, we need a highly specific modulation targeted at only one muscarinic receptor, ideally, and maybe even tissue-specific modulation. But here we see cross-sections of 4 muscarinic receptors, their structure, with electrostatic potential on them. In a yellow ellipses, there is a so-called orthosteric binding site where acetylcholine binds. And as you can see, this site is absolutely identical among receptor subtypes. This means it's virtually impossible to get ligand that binds solely to the orthosteric binding site and is selective among subtypes based on their affinity. So what we can do on the top left, with Q and B, we have a M2 receptors with bound Q and B, which is non-selective antagonist. As you can see, it binds to the orthosteric binding site. Beneath that is the M2 selective antagonist, AFDX384. And as you can see, it somehow interacts also with the receptor outside of orthosteric binding site and gains some selectivity. So to get antagonists, we somehow have to reach out of the binding pocket. But here we have a M2 receptor is bound hyperoxal. That's muscarinic superagonist. And what we see, that upon activation of the receptor, the binding site closes and agonist cannot reach out of the orthosteric binding site. So what we can do, because we cannot get the selectivity from the extracellular part of the receptor, it's allosteric modulation. This compound binds to the extracellular part of the receptors and increases affinity and efficacy of hyperoxal. So it's a so-called positive allosteric modulator. So to somehow improve the signaling, we need positive allosteric modulators. And interesting thing is that we can have also negative allosteric modulators. So by which we can modulate. So we can use this allosteric binding site to modulate muscarinic signaling either way, either facilitated or inhibited. So that's all from me as a brief introduction. And so if you are interested how we do this basic neuropharmacological research, you are welcome in the afternoon in our laboratory. Thank you.
So good morning, my name is Helena Janičková and I am also coming from the Department of Neurochemistry, but I will present a slightly different topic, well actually quite a different topic, which is titled Nicotinic acetylcholine receptors expressed by specific neuronal populations. I realize this may sound a little bit abstract, but in the following 5 to 10 minutes, I will try to show you that what the topic is about, and it is actually quite relatable. So in order to understand what nicotinic acetylcholine receptors expressed by specific neuronal populations are, some of you maybe need a crash course on nicotinic acetylcholine receptors. So they are ion channels, they are pentamers consisting of different alpha and beta subunits, and they are permeable for calcium, sodium, and potassium ions. They can be located presynaptically, postsynaptically, or extrasynaptically. And in these locations, they control the release of different neurotransmitters. They control neuronal excitability and also neuronal plasticity. I just told you the nicotinic acetylcholine receptors are pentamers composed of different alpha and beta subunits, and which means because these potential combinations of these subunits can be quite numerous, it means we can have multiple different subtypes of nicotinic receptors. And I'm sure most of you know that the neurons in the brain are not just neurons, but there are tens and likely more different populations of neurons that differ based on their morphology, electrophysiological properties, and gene expression. So if you combine these two complexities to, different subtypes of nicotinic receptors and different populations of neurons, you can create even more complexity, which brings interesting questions. Like, for example, do different populations of neurons express specific sets of subtypes of nicotinic receptors, and how these sets of nicotinic receptors affect their functions? Or, for example, do different subtypes of nicotinic receptors expressed by different, do the same subtypes of nicotinic receptors expressed by different neuronal populations have the same or different functions in the brain, in the neurons, and different or same effects on behavior? Anyway, the take-home message on nicotinic receptors here is that they are widely expressed throughout the brain, that they are involved in the control of fundamental brain functions. And in order to use them as a target in therapy, we can't treat them as a monolithic blob, but we need to investigate their expression and function in specific neuronal populations. And currently, our favorite neuron population we are interested in are cholinergic interneurons in the striatum, which is this blue circle. And I think right now you can take in another crash course. So I will just show you, not this direction. They look a little bit like this. And you need to believe me, they are associated with a broad range of different types of behavior and also with a broad range of neuropsychiatric disorders. So our research goals when studying nicotinic acetylcholine receptors in striatal cholinergic interneurons are basically as follows. We want to map the expression of nicotinic receptors in the cholinergic interneurons. We want to examine how these nicotinic receptors in these neurons influence their activity and also the activity of the local network and eventually behavior. And we want to explore approaches that could us allow to target these nicotinic receptors more selectively in the cholinergic interneurons in the striatum. So to reach these goals, we at first used fish, and we established that cholinergic interneurons in the striatum commonly express beta-2 nicotinic subunit. So therefore, we created a genetic mouse model where we deleted this beta-2 nicotinic subunit in cholinergic interneurons. And then we tested this model behaviorally and biochemically. So if you are quick enough to grasp or to navigate this slide, because we don't have so much time to spare, I am afraid, you can appreciate that mice with the deletion of beta-2 nicotinic subunit in these cholinergic interneurons show changes in behavior, specifically in the social and anxiety-like behavior, and they also show changes in the activity of striatal neurons. Specifically, they show decrease of activity of cholinergic interneurons, and on the other hand, in contrast, they show increase of the activity of the general striatal population of neurons. So when we showed this, we wanted to take a next step and to investigate what is the effect of the beta-2 nicotinic subunit or the nicotinic receptors containing this subunit in cholinergic interneurons. What is the effect on the electrophysiological properties and the firing of cholinergic interneurons in the striatum? And for this, we have developed a new genetic mouse model. It's a Shed-Cree beta-2 flox-flox crossbred, and we also established a collaboration with the electrophysiological laboratory of Professor Schiffman in Brussels, where our current PhD student, Amanda, is currently learning and recording cholinergic interneurons in the striatum. And now I am getting to the main point of my presentation, because while Amanda is struggling with patch clamp in Brussels, we also need to do a lot of work in Prague. So we are currently looking for a master or even a bachelor student who could help us to characterize our new mouse model and who could help us to answer some of the following questions or even some other related ones. So for example, using the immunofluorescence, you could answer the question whether the Shed-Cree expression in this knock-in Shed-Cree mouse line reflects the true expression of Shed in the mouse brain, Shed being choline acetyltransferase. By doing image analysis of some previously acquired samples, you could answer the question whether to what extent other cholinergic neurons other than cholinergic interneurons in the striatum also express beta-2 nicotinic subunit. Also using quantitative PCR and behavioral tests, you should answer the question whether the Cree insertion into the Shed locus has any effect on Shed baseline expression and behavioral phenotype in the control mouse line, and also to establish whether there are any behavioral changes in the Shed-Cree beta-2 flox-flox mice. Or potentially using stereotaxic injections and AAV vectors, trying to create even more specific model with a more specific deletion of beta-2 subunit exclusively in the cholinergic interneurons. So although the topic is relatively recent, I've already collected a small set of references I can provide, and although these students are not named in full, I think all of them would be happy to testify that I am a decent supervisor. And I'm showing this so you can appreciate the approximate scope of the possible topics for your thesis, either master or bachelor. And you can see that in particular for the bachelor thesis, if the students are interested or they are supposed to write only theoretical work, only theoretical-based thesis, the scope of topics related to nicotinic receptors can be really quite broad because they are literally everywhere, and I'm always interested to learn something new about them through your thesis as well. So I would like to thank you for your attention, and if you are curious or have any question, please drop by in the afternoon or just send me a message. Thank you.
Thank you very much. And next presentation will be from the Laboratory of Molecular Neurobiology, given by the head of this laboratory, Martin Balaštík.
Okay, so good morning, everybody. My name is Martin Balaštík. I'm the head, as you heard, of the Laboratory of Molecular Neurobiology here at the Institute. And I'm going to be telling you in next couple of minutes what we are doing in my laboratory, what is the subject of our work. and methods what we use, and then we can discuss even more later when if you would come to the lab. So let's start with the human nervous system with the brain. You may know that human nervous system or brain is thought to be consisted of 86 billion neurons that are forming 10 to the 14th of synapses, which is a number that is very hard to imagine. You can think about it as it's like if all the people in this planet and 10 other planets would get connected, 10 connections, 1,000 connections per person, and then you just wrap it in 1 kilo of matter that you have in your head. Very important about those connections is that they are not random. They are very precise. So some processes from let's say one area, like as a lateral geniculate nucleus, is reaching, for instance, here primary visual cortex, and not only that, it just grows through the various layers, sends out collaterals, and makes connections with specific neurons. And it has been well established that changes in the number of synapses has been related to various neurodevelopmental disorders, like autism spectrum disorders appears to have increased number of synapses in adolescents, while schizophrenia has reduced number of synapses. And obviously, various neurodegenerative disorders like Alzheimer's disease in the adulthood or in the aging has also a drop in the number of synapses. Okay, what we are interested in is what are the mechanisms that are generating the specificity of those synapses and the number of connections, etc. How is that achieved? You can think about it that in the development of human as well as mouse cortex, there are several critical milestones that have to be reached in order to generate the proper functioning cortex, which is a proliferation of the progenitors, neurogenesis through the asymmetric division, then migration of those progenitors in formation and their localization, forming the layers, as well as then differentiation and forming the connections. And defects in any of those steps have been linked to several neurodevelopmental disorders. I was already mentioning autism spectrum disorder, but there's also schizophrenia, Fragile X syndrome, epilepsy, etc., etc. And all those milestones are very different indeed, but they have one thing in common. They are mechanical processes, and they need some motor system to carry them on. And obviously, in the cells, in the neurons, this is a cytoskeleton. And it appears that it's particularly critical or dependent on microtubules, because if you consider various gene variants or mutations that have been associated with several of the most common neurodevelopmental disorders like lissencephaly, microcephaly, etc., you will find many isotypes of tubulin mutations or microtubule associated proteins. And then what we are doing in my lab, we in a condensed way, is we are studying like how different changes of post-translational modifications in microtubules affect various microtubule associated protein, their binding, their activity, how also different post-translational modifications of these microtubule associated proteins, their alternative splicing, as well as conformational changes affect activity of those proteins, and the whole kind of complex affect axon guidance and synapse formation or refinement, and brain development and various neurodevelopmental disorders. And if I talk about microtubule-associated proteins, just for you to mention, this is a rather large group of proteins that is defined rather just by close proximity to microtubules. There can be some motor proteins, microtubule-stabilizing proteins, destabilizing, severing proteins, or end-tracking proteins, as you can see here. What we are studying particularly are those proteins that are binding the tips and just affect the growth or collapse of the microtubules in the growth cones, as well as we have now a project that is focusing on the motor proteins that are basically being distributing cargoes across the axons. So this is something that actually, if you come to my lab, we're going to be discussing more. Now, just to tell you the methods that we use in the lab, we are generating knockout mice. We have already generated a couple of unique knockout mice that we further analyze. If we want to mutate or tag only specific neurons in the developing cortex, we use so-called in utero electroporation, where we inject some DNA plasmids into the ventricles of the developing cortex, electroporate them, and just generate mice that has only some of the neurons tagged or mutant. As you can see here, this is a cross-section of a mouse brain, and here those are fluorescence neurons that were electroporated, and you can track their projections, their axons going to the contralateral cortex, as well as their dendritic arbors, and this is in the distal contralateral cortex. And we are using also organotypic brain slice cultures where we analyze migration of some electroporated neurons inside the slice towards the upper layers. And we can just nicely see how various mutations affect behavior of these migrating neurons. We are injecting and basically tracing some neuron projections within the cortex using fluorescent dyes. Here is a dye eye injection. We use gene GAN in order to specifically label the whole kind of neuron with a fluorescent dye within a brain slice, and then we can just analyze really in a huge detail in how the arborization is changing, how their connectivity is changing, etc. We are using primary neuron cultures or explant cultures, so you can learn that in our lab too. This is actually what we do a lot, as well as primary neuron cultures in the microfluidic chambers, where basically we can separate and divide their proximal and distant projections, where the dendrites of the axons are, the dendrites of the neurons are here and in one chamber, and you see only axons growing to the distal part, and we can just analyze exactly what is the effect of our various treatment into those neurons. In collaboration with Zdeněk Lánský's lab, we are testing what the genes that we work with do in vitro. So we are doing so-called in vitro microtubule polymerization assays, where you have like the little stub of microtubules, and then in vitro you can see how the protein that is binding, you see it in the green, how it promotes polymerization of microtubules or not, or just induces collapse of this protein. I mentioned that we are using multiple microscopy techniques of confocal TIRF microscopy, light sheet microscopy, just to see specific effect on the cytoarchitecture of those mouse models that we generate, etc. Those are some of the recent publications that we have gathered using our techniques and models. And I have to thank people in my lab that are doing the work. This is the current setup, the collaborations within the institute, as well as our great collaborators that we have at the Institute of Curie Technical Institute, Technical University in Munich, or Tel Aviv University. And if you are interested in what we are doing, just definitely come to my lab. We are interested in master and bachelor students. So you could just really learn those techniques. And we have several projects that are related to the axonal transport in using those microfluidic chambers, etc. So if you're interested, stand by and we can discuss it more. Thank you.
Thank you, Martin. And the next presentation will be from the Laboratory of Biological Rhythm, given by the head of this laboratory, Alena Sumova.
Hello, everybody. So I'm Alena Sumova, head of the Laboratory of Biological Rhythms here at the Institute, and I would like to advertise a topic for a student project entitled Development of Circadian Clocks in a Fetal Brain and Their Sensitivity to Maternal Stress. So I need to first briefly overview what we are studying. So our studies belong to the field called chronobiology, which is a field studying temporal regulation of biological processes. And specifically, we are interested in circadian rhythms. These are driven in our body by endogenous clocks located at various parts of our clock. And these clocks drive rhythms with a period very close to 1 day. This is why we call them circadian rhythms. And as we know that these clocks are really widespread in our body, this field is really multidisciplinary. And significance of this regulation for our health had been recognized by awarding Nobel Prize for Physiology and Medicine in 2017. So now we really know it's very important. So the circadian clocks, as I said, are widespread and they organize many physiological processes in our body. So they not only tell us when to go to bed, when to wake up, and to ensure that this occurs at appropriate time of the day, but they also organize all other processes in alignment with these cycles. So they drive rhythm in our metabolic processes, cardiovascular functions, hormonal levels, and many, many others. And the important thing is that due to the regulation, all the rhythms are well synchronized. And we know that disruption in this synchrony as often occurs due to our unhealthy and irregular lifestyle is associated with many serious disorders. So these disorders are often associated with our unhealthy lifestyle, and we know that disrupted rhythms are involved. So what we are studying, we study circadian clocks and all levels of the organization. So mainly we are interested in a center clock in the brain, which is located in a region called suprachiasmatic nuclei. This center clock is the only clock in our body which receives information about external light-dark cycle and synchronizes with external time. And then it drives directly various systemic rhythms, as I mentioned before, but like sleep-wake cycle, for example. At the same time, it sends signals to clocks in other parts, either in other regions in brain or in various organs to tell them time of a day. So this is very important clock. And we study organization and clocks in all these levels of the hierarchy. So to study the clocks, we can either follow the clock-driven rhythms, very easily monitor sleep-wake cycle in our animals in laboratory or in human studies, follow activity and rest as an output of the clock. But what we want to see is to look directly in the molecular mechanism generating the rhythmic signal in our body. So we can now follow, in fact, circadian clocks ticking in various parts of our body in real time. So we can follow clocks in various regions in the brain to study neuronal regulation, circadian regulation of neuronal processes, or in these tissues. So how can we do it? It is because we have a transgenic mouse model in which one of the clock component, molecular component of the clock mechanism, HER2 protein, is fused with a reporter. And reporter is luciferase, which can be easily monitored as a bioluminescence. So what we do is that we prepare explants of the region of interest, either from the brain, like in this case, the piece of a brain containing the center clock, and we culture it ex vivo in a culture dish. And then we use various devices to monitor clock-driven bioluminescence. And we can do it for many days as long as the tissue is alive in culture. And from that recording, we can analyze the properties of the clock, how fast it is ticking, what is the amplitude, what is the phase of the clock, etc., etc. And we can do it at a tissue level as well as at a single cell level. So this topic I am advertising here today is related to our long-term interest in the issue how the clock develops during ontogenesis. And we already know that this development of the clock rhythmicity is correlated with morphological development of the structure of the suprachiasmatic nuclei. And we also know that the development is affected by maternal clock. Maternal circadian system has impact on the process. But still many questions remain to be answered here. So we still don't know how exactly the SCN clock develops. We also don't know what maternal signals affect the developing clock. And I just show example of results of one of our recent study in which we address this issue as just to show how our studies look like. So we were interested to identify genes which are rhythmically expressed in a fetal SCN, and to identify those genes in mothers in which their clock was intact, so these were control mothers, and to compare it with gene expression profiles in mothers in which we removed the SCN surgically and imposed them rhythm in feeding and fasting. Importantly, the fetal SCN was collected at the time before endogenous clock, fetal clock developed. So the idea was to identify rhythms which were driven by rhythmic maternal signals. And using the biostatistic tools, I will not go into details, we were able to sort out these genes, rhythmically expressed genes, into specific subgroups, genes which were rhythmic only in the controls, genes which were rhythmic only in those mothers in which we removed the SCN, and the largest group were genes rhythmic in both groups in synchrony. And this is what we were interested in because those genes, in fact, were driven by rhythmic maternal behavior. So our data showed that the rhythmic maternal behavior is really very important for this rhythmicity. And when we identify those genes, we found that they are tightly interrelated into the neuronal development of the structure and also into inter and intercellular signaling. So these results clearly stress importance of regular daily regime during pregnancy. And they also point at the fact that preterm delivered babies are prematurely devoid of those signals, which really stresses importance to study these pathways. So now we are following up to understand how the fetal clock responds to maternal stress. So this is based on our recent data where we found that indeed the fetal SCN contains glucocorticoid receptors. There is a rhythmic expression in level of those receptors, and that the fetal SCN responds to injection of synthetic glucocorticoid dexamethasone to pregnant mothers. So it responds by change in electrical activity, neural activity. So we tested how this drug, this synthetic glucocorticoid, affects the clock in vitro and found that indeed explanted fetal SCN clock responds by increasing amplitude, changing period, and importantly, also shifting at specific time of a day, which could be blocked by specific antagonists of the glucocorticoid receptors. So now we want to follow up on this data because we really need to understand molecular mechanisms which connects the glucocorticoid receptor activation and the molecular circadian clock. So this is the topic of the project I am advertising. So here is the list what we can offer to you. So we can offer you a really fascinating and multidisciplinary topic, which are the biological rhythms. They are currently attracting attention of almost all biomedicine fields because of their widespread significance. We also can offer you working at a really internationally recognized laboratory and in a young, friendly, and open mind and international environment. And we can promise to train you with a wide range of techniques, behavioral, molecular techniques, just to repeat that we do all these omics, transcriptomic, proteomic, metabolomic analysis, real-time recording, gene expression, or AAV transfection, microscopic techniques. I'm not listing all of them. So what we expect from you, just be really enthusiastic and highly motivated. For candidates on PhD project, you should already have or receive in soon master degree in a relevant field. And we also would like you would be able to communicate in English because of our international team, and also if you have any previous experience with any of the technique, this would be advantage, but definitely not required. So that's all. This is our team and our international collaborators, and all the team is now looking forward to invite you on the board. Thank you.
Thank you very much. And now I would like to welcome here 3 representatives of our PhD students that will tell you more about their studies at the Institute. Alexandra, Teresa, and Marco are highly motivated students. I am glad to have them here because I am also responsible for all outreach activities, and all of them they help me always to present the institute for the public. So who is the first? Marco?
Hello, everyone. So right now we will talk a little bit about our experiences here as PhD students. So as you can see in this picture, there are many of us at this institute, but 3 of us right now will tell you what we consider the most important things that we learned so far from our work. So firstly, we will introduce ourselves a little bit. So my name is Marco. I'm a 4th-year PhD student. I work at the Department of Adipose Tissue Biology, and I'm enrolled at the first medical faculty as a PhD student. So the topic of my research is metabolic effects of exercise with a focus on adipose tissue function. So basically how exercise helps our fat tissue to be healthier and function better.
All right. Hello, everybody. My name is Teresa, and I'm doing my 3rd year, or I'm in a 3rd year PhD, and I'm working at the Department of the Biological Rhythms. So you previously heard my supervisor, Alana. My research topic is focused on a characterization of the circadian clock in the choroid plexus, which is like a tiny structure in a brain ventricles, synthesized cerebrospinal fluid.
Hello, everybody. I'm Alexandra, and I'm also a PhD student. Now I'm currently doing my 2nd year at the Department of Cellular Neurophysiology, where we study ion channels called the TRP receptors. You've heard of or about one in the first lecture, actually. I, from the beginning, would like to mention how I got here and maybe encourage you a little bit, because in this lab in which I am right now, I came here to do my master's, and then I really liked it, so I decided to continue and do my PhD. I remember when I looked at the list of you and I saw that most of you are studying the molecular biology at the Faculty of Science of Charles University, which is the program which I studied also. And I remember that I saw an advertisement for open position for a student, and it was on a Facebook group. So I sent it a Facebook message, which was not official enough because I did not get my reply. I don't blame the person because she did not actually saw the message. And I remember that at that time I was so insecure and I thought that I have no experience and why would they want me? So I did not push it. But so then I did my bachelor thesis at the faculty. And after that, I still had this lab in my mind. So I sent official email. I actually got a reply to that email. They told me to come to the lab to see what they do and how it looks. I came and I kind of stayed since. So don't worry, you don't need to have any experience. I think that you heard that before. The only thing you need you want to work. You have to be enthusiastic about it and you gain the experience, so don't worry. And now Marco will tell you what he thinks is important or what you maybe should think about before you join.
So I wanted to start in a little bit philosophical manner because I felt like this is the most important advice that I would give someone who asked me like what is the most important for this job to be successful and to be happy with what you are doing. So I want you to think what is the similarity and the difference between science versus art. So firstly, the difference is in art, the most important things are your feelings, how you perceive things and what you feel inside. In the science, the most important things are data and objectivity. So science in the science, data are more important than feelings. But in both fields really demand your creativity, to think creative. So in the science, you really wanna why is it important that you love your topic so much and the field that you are part of? You really want to have that intrinsic urge to ask questions and to solve problems. So if you imagine some artist like Mozart, for example, he was creating music, but was it the obligation? Was he pushed to do it? No, it was just an enormous amount of creative energy inside him that wants to explode out. So we want really to find a field that you're going to feel very similar in the science, that you're going to go home and you want your job to be part of yourself still when you are not here. But you don't want it to be pushed to do it. It has to be natural part of yourself. It's because sometimes in the science, you're going to get the best ideas when you are not here at the work. Maybe when you are drunk at the party at 3 a.m., you're going to get fantastic idea what you should measure very soon. And, things are going to be much better in this case. So you want to ask yourself, do you want to be a researcher or a technician? So when you do PhD, you are trained to be a researcher. And that means having creative mind, thinking, solving problems, and so on. Otherwise, you're going to just be a technician. If you don't love what you do, you're going to be told by your boss to measure certain things. You're going to measure them. Then your boss will look at the results and tell you what to do next. So you're just going to do technical things and not anything else. So now I'm gonna show you some nice experiences that I have, and before that, also before you start PhD, make sure to visit the lab, but not just today. Find what lab interests you the most. Send the email to the boss, and they will definitely reply you, so you can work a little bit with them to see if you like what you do and if you like the team and if they like you. So, for example, before I started PhD, I had a 2-month internship, and I made sure that I like what they do and that I like them and that they like me. So now, after that, our collaboration is pretty good. And also, as a PhD student, you will have the opportunities to travel, to meet the experts from all over the world in your field. So, for example, these are pictures from my last conference that happened in January this year. So it was in the United States. The title of the conference was Exercise Biology, as you see here. So I met all the leaders in the field of exercise physiology, had the opportunity to talk to them and so on. So you will definitely have nice opportunities to travel, to do stuff that are fun, but the most important thing is what I started with. So make sure that you are really about that topic that you're gonna work on. Thank you.
Okay, so maybe I would like to continue when Marco started. It can sound a little bit poetic, but really don't be afraid to follow your dreams and make big changes. Because as you can see, my path was really long and really different. I started my studies at Palacký University at Olomouc, like experimental biology / chemist. At the beginning, I focused mostly on studying plant hormones, and after that completely moved to the organic chemistry and focus on a synthesis of the new potential drug for jet lag therapy. And this was the moment when I said, okay, circadian that can be maybe interesting. And I realized that maybe I would like to focus more on neuroscience and brain research. So my advice for you, or from my experience, sometimes it can be really hard to make the decision and make that change, because you can feel like, okay, I start research on that topic, so maybe I can continue with it because it's like more easy, I can have more data or something like that. But it's really important to find something you really like, so you don't just do something so you can do something. Just do something you really love. And don't be afraid of changes. It's fine that you can start from the beginning over and over. And also from my experiences from here, from the Institute, as Marco said, there is the opportunity to visit lots of conferences, summer schools, internships, and workshops. For example, from me, it was really interesting to visit a workshop for neuroscience at the Nenský Institute, organized by Dirk Schubert. Because it's like you are reading the work of the really most famous scientists, and after that you have the opportunity to meet them and chat with them, and you can also have a collaboration with others and meet the new people, make your network. And I think that I'm really glad that Institute gave me that opportunity to change like my field and focus on my favorite research and favorite topic. That's all from me.
So now you back to the conferences, which Marco talked about a little bit. We are here 3 PhD students, so now you are maybe thinking, I'm not there yet, so am I able to go to some conference? Don't worry, because I remember when I was doing my master's, I still had this opportunity, so I don't think it's something which you can find in some labs. I think it's if you would be here, you would have the opportunity. And maybe if you're lucky as I am and someone in your field wins a Nobel Prize, you have a really big chance of actually seeing them or meeting them in person, which is nice. And if you have your little scientific crush on them, it can grow even more. But jokes aside, what's another thing that I really like about the whole study process, especially here, is that you can, while you study, you can apply for some form of student grants and research fundings. It's here, your supervisors would help you to write the application because they do have a lot of experience with writing them. I think it's, even though you don't actually get the funding, I think it's a brilliant opportunity for you because you actually get to see how the fundings, how it can work. And if you get it, I believe you become more independent. You actually will be responsible for something. So definitely try it. Don't feel intimidated by it, like I did at the beginning. For example, I do believe that there is plenty of agencies or programs where you would be able to apply for some grants. I have personally experience with the Charles University Grant Agency, for which you can apply while you are a master's student or when you are a PhD student. Me and my team, we actually won the Rector's Award for the best project of last year. And I think that the whole thing actually reflects how the supervisors here and the teams can be really great and brilliant. So don't be afraid, ask your supervisors, because here are people who actually have time for you. I believe that they would be interested in teaching you and showing you how to do it. They will have time for you and you would be included in the research, which is one of the things that I like about this place so much, is that you don't be doing something just for the sake of doing your bachelor thesis or master thesis. You would actually be included in the research as it is.
Okay, thank you. And so I can continue with this. As Alexandra said, that it's really amazing that you are not just sitting here writing or reading, but you are actually can be involved in a research. And yeah, it can start like the simple pipetting, but if you are successful, it can kind of end like the most exciting thing in the research, and it's a publishing of your own scientific paper. I mean, during your master thesis, it's not really often that you will be like the first author, but it's happening quite often that you can be a co-author. And I think that we can close it here if you don't want to add something. And because we are a little bit light, maybe we can leave the discussion in a coffee room, and we are really open to your questions and everything, and we can discuss everything there. Thank you.
Thank you very much, all of you. It was a nice overview or the view into the studies, and I think you did it very well. So welcome to the second part of this morning session. There will be a little change in the program because Ivana Vaničková from the experimental hypertension left. So I will start with my presentation, which is the Laboratory of Membrane Transport. I am a vice head of this laboratory, and I would like to tell you more what we are studying. As you heard from the director, the research of the Institute concerns studies from the molecular levels up to the whole organisms. And from the title of our laboratory, you can see that we study membrane, and we belong at the bottom of this pyramid because cell membranes are selectively permeable, and transport of compounds through the membranes is highly important for all life. And we are interested in various transporters that are membrane proteins that transport not only compounds, but also signals across the cell membranes. And it is necessary for the uptake of nutrients, efflux of waste compounds from cells, for the communication with the environment, and for the ion homeostasis, as we will see in a while. And we are a unique member of our lab is the unique member of the institute, because we are the only one who use as a model organism of a eukaryotic cell, yeast Saccharomyces cerevisiae. So we don't work with animals. And the yeast Saccharomyces cerevisiae is a unicellular eukaryotic microorganism useful not only for the production of alcoholic beverages or bread, but we use it to explore molecular aspects of various fundamental cellular processes that are very similar in yeast cells as in higher eukaryotes. The work with yeast is little bit easier than with animal cells and animals in general. Yeast are rapidly and cheaply, more cheaply cultivated, and we have enhanced various highly developed methods of molecular biology and genome engineering. And we are using yeast also for the expression and for the study of proteins from higher eukaryotes to explore, to find out more information about about these human proteins, for example, and then you can even mediate the simplified model of human disease, which is in the modern time called humanized yeast. As concerned the methodology in our laboratory, we use a wide range of methods that concerns bioinformatics, molecular biology, biochemistry, microbiology, and physiology and biophysics. And we have a collaboration in the Czech Republic and also abroad. And with the laboratories that concerns bioinformatics in Israel, for example, or the advanced optical microscopy in Germany. And this optical microscopy laboratories that are specialized on these advanced techniques are present also in the campus or in BioSef. This is the current team of our laboratory. And you can see that there are two positions, at least two positions for one PhD student and 1 to 3 master science or bachelor students. So you are really welcome. And I will present you one of the topic that we offer for students that concerns cations as a key elements for life of eukaryotic cells. Because for the long time we studied the regulation of monovalent cation homeostasis in cells. And I am sure that all of you know that the most important monovalent cations are protons, sodium and potassium. Protons, as their concentration determines the pH of the environment, which is not unique or unify even within the cell. Sodium as well as protons are very important for other transporters because their gradients across the membranes is necessary to establish the electrochemical gradient across the membrane. But the high concentration of sodium inside the cells is toxic for cells, so it must be eliminated. On the other hand, potassium cations must be accumulated in cells in high concentrations. And this phenomena is preserved from bacteria up to animal cells, and any disturbance in this cation homeostasis results in pathology and various diseases. So the pathology and the malfunctioning is observable also already on individual cells. And then even worse is in the whole organism. So, and to maintain cation homeostasis, cells possesses in membranes various types of cation transporters like channels, ATPases, symporters, and antiporters. And this is the topic what we are interested in. And I would divide our interest into 5 points. First, that we study the structures and functions of cation transporters from yeast cells. Here you can see the model, the picture of yeast cells with all known cation transporters. And recently we characterized the structure of one of the unique member of yeast transporters that could be also a possible antifungal target. We also, and it will be the main topic of the PhD study, like to study organization of transporters within the plasma membrane, because they are expressed quite at very low level, and about the organization, how they, if there is some organization, it is not known. And this is what we are interested in now. So someone who would like to become expert in microscopic techniques is really welcome. We also study the proteins that are important for biogenesis or degradation of membrane proteins. An example you can see here that for the sodium proton antiporter, which is normally localized in the plasma membrane, the lack of one very small protein in the endoplasmic reticulum results that it's stacking in ER and not a good localization in the plasma membrane. So these proteins are interesting for us. We also search for new regulatory mechanisms that regulate transporters at the molecular level. And here again, you can see our recent study that we identified an interaction between NH, this sodium protein antiporter, and the 14 free proteins. And we realized that this interaction is important for the inhibition of the activity of transporter. And finally, very recent work from our lab is that we characterize human transporters in yeast cells. And by this approach, we can find many important information. As you can see in this picture, we identified residues and regions that are important for the activity, and also we identified the region where the inhibitor of the molecule is binding. So all these studies are at the molecular level and the one cell level. And the next two projects for bachelor and master science degree will be presented by my colleague.
Good morning. I will tell you something more about these two topics. The first one is the fluorescence probes for the study of cell membranes. The proper lipid composition and function of cell membranes is essential for the life of all organisms. Fluorescence probes are useful tools for estimating membrane fluidity, membrane potential generated by ion transport across the membrane, molecular interaction within the membrane, and many other things. The first fluorescence assay is DCC3 assay usable for measurement of plasma membrane potential. It was developed by Dana Gašková at the Faculty of Mathematics and Physics of Charles University, and it is based on the DCC3 redistribution potentiometric fluorescent probe. It is a lipophilic cationic dye which easily passes through the plasma membrane and accumulates in the cells in response to the inside negative membrane potential. The binding of the probe to the cell components is accompanied by changes in the spectroscopic properties. The spectrum of the bound probe is shifted towards longer wavelengths, and the intensity considerably increases. So we monitor the time evolution of the lambda max position after addition of the probe to the cell suspension, the so-called staining curve. The probe is also a substrate of two major MDR pumps of Saccharomyces cerevisiae PDR5 and SMQ2 and Candida glabrata CDR1. Therefore, we can see differences between these two staining curves according to the presence or absence of CDR1. The big advantage of this method is that enables us not only measurement or detection of pump inhibition by some antifungal drugs, but also measurement of effect of these drugs on the integrity of plasma membrane. You can see it on the model experiment where the cells were pretreated with various drugs before the addition of the probe, and the staining curve of the permeabilized cells reveals very rapid shift, which is typical for cells with disrupted or fully permeabilized membranes. The second method will be completely new method developed by the student based on the fluorescent probe Lordan and suitable for measurement of membrane fluidity. The Lordan is a lipophilic and dye which is anchored in the plasma layer due to its lauric acid, and fluorescent naphthalene residue is located near the glycerol backbone of the lipids. Lordan enables to measurement so-called generalized polarization and is very sensitive to penetration of water into lipid bilayer. So you can see that with increasing temperature, increase also fluidity of the membrane and change the Lordan spectrum. The last technique will be new method based on the body pipe probe, suitable for monitoring lipid peroxidation. And this probe incorporates into lipid membrane due to its fatty acid, and upon oxidation exhibits a shift in its fluorescence from red to green. Lipid peroxidation is oxidative degradation of cellular lipids by free radicals from reactive oxygen species that can attack double bonds of phospholipids or unsaturated fatty acids. So the lipid peroxides can damage the membrane and eventually cause the cell death. And the second topic is about the transporters involved in multi-drug resistance in Candida species. Candida species cause serious life-threatening infections. Their mortality is quite high. They are able to colonize various areas of the human body. But the number of clinically useful antifungals is quite limited. And treatment is complicated by the emergence of multi-drug resistance caused by membrane transporters, which decrease the concentration of drugs under the sufficient level, which is able to kill the cells. According to the source of energy, we can divide MDR transporters into two groups. ABC primary active transporters use ATP as a source of energy, like this human P glycoprotein, or secondary active transporter use the gradients of protons or sodium cations as a source of energy. We will focus on the second most spread Candida glabrata in our study, and its transporters ABC, CDR1 and CDR2, and 9 proton drug antiporters. We will study substrate specificity of all these transporters. We can use a conventional biological method like this diffusion test, or we can use disease-free assay, as I mentioned previously, because this probe is a substrate of the CDR1. And we will also develop a new fluorescence technique based on these sterile pyridinium compounds, because we expect that some of these 8 compounds can be substrate of these transporters. As you can see from this picture, really compound 8 is a substrate of CDR1 and CDR2. And what is also nice that these compounds have a quite big Stokes shift that is really advantageous feature for microscopy techniques. And here you can see illustration from compound 5 as an application as a vital dye, because it accumulates in the dead cell compared to the live cells. So if you are interested in our topic, you can contact us or visit our laboratory during the afternoon. Thank you very much for your attention.
Thank you. So if you are interested in cations and membranes and methods that are used for the characterization of cells, you are really welcome into our lab in the afternoon. And now I would like to pass the word to the Laboratory of Experimental Hypertension and to the head of this laboratory, Ivana Vaničková.
Thank you very much. Hypertension is associated with many diseases and leads to high morbidity and mortality in the population. Therefore, its study is necessary, and our experimental studies are based on the analysis of various organs. You may probably know that various organs or tissues are affected: atherosclerosis in vessel, stroke in the brain, retinopathy in the eye, and also the important diseases such as renal failure or myocardial infarction are associated with hypertension. Our group is focused both on the basic research, meaning the evaluation of mechanisms of blood pressure control, and the role of oxidative stress and mitochondria in these processes, as well as the contribution of sympathetic and parasympathetic nervous system in these processes. Moreover, we are also involved in part of the applied research, and our topic today is the role of glyphosines in the treatment of non-diabetic diseases. And we also, a part of our group collaborates with the Institute of Organic Chemistry and Biochemistry, and is involved in the research of the role of neuropeptides in food consumption regulation. So our studies are based on animal models and basically on rat models of hypertension. Sorry. So the main there are two possible ways or parts in which hypertension is studied. So one are the genetic models, for example, spontaneously hypertensive rats, in which the sympathetic nervous system is highly activated. The alcohol-sensitive rats are the rat strain, which is dependent on the intake of sodium. In sensitive animals, hypertension develops, while in the resistant rats, they do not develop hypertension even under high salt intake. And there is a special monogenic model of hypertension, which is determined by the activated renin-angiotensin system, and this is REN2 transgenic rats. We are also able to induce hypertension experimentally, and these are these models which are mainly used in our labs. And this is the reduction of renal mass and renal vascular hypertension. And the first one is shown here, where one kidney is fully removed and the poles of the remnant kidney are removed too, which leads to the hypertension development and finally also to chronic kidney disease. And similarly, we can induce chronic kidney disease by the insertion or clipping of one renal artery, which induces the collapse of this clipped kidney and the enlargement of the other kidney and the activation of renin-angiotensin system. We can also use the models of heart failure. I have already mentioned these two models. And when spontaneously hypertensive rats are on a high-fat diet, the heart failure model, which with systolic and diastolic dysfunction develops. Another possibility how to induce heart failure is the placing of dull salt-sensitive rats on high salt intake for a longer time, and they also are able to develop signs of heart failure. Concerning our methodological approach, we are working from the whole animal to the molecular biology techniques. And in the afternoon session, you can hear something more in detail about the methods of blood pressure measurement used in our lab. The most important or precise method is the telemetry blood pressure measurement, with which we can also analyze other parameters such as heart rate variability or blood pressure variability. We can also measure blood pressure and responses of various drugs in conscious animals during the direct blood pressure measurement using cannulation of carotid and jugular vein. Another possibility which we are using is the annulation of lateral brain ventricles and infusion of drugs that also somehow influence blood pressure. On the organ level, we can use perfused vascular beds or the properties of vessels. We can analyze on using isolated conduit and resistant arteries measurement with Mulvaney's myography. And as I already mentioned, we are using different immunohistochemistry method as well as molecular biology methods. So the title of my PhD project is the mechanisms of glyphosine action in the treatment of non-diabetic models of cardiovascular diseases. The glyphosines are the inhibitors of sodium glucose transporter, and originally they were used for the treatment of diabetic patients. But later on, they were shown to be able to somehow affect other parameters also in non-diabetic patients. So they induces natriuresis, leading to lowering of proteinuria and lowering of blood pressure. And through increases in glycosuria, there is a substantial caloric loss and major metabolic effects. Moreover, they are able to also to influence cardiac function, and altogether they have important cardiovascular benefits for patients and also for non-diabetic patients. So these are only mention the big clinical trials which were performed in thousands of patients, showing that they substantially improved not only cardiac function but also renal function. But the question is whether they perform the same effects under non-diabetic conditions, and what are the mechanisms for these effects. And I'm showing here the results of our previous studies with three different hypertensive models. And you can see here that in all these models, the effects on body weight and visceral fat mass was very effective. But based on the type of the strain used, there were other parameters that were substantially affected. Blood pressure was decreased in renal-transgenic rats, while hepatic lipid metabolism was improved in the metabolic model of hypertension, and in spontaneously hypertensive rats with increased in expression of CRP, the inflammation and oxidative stress were substantially improved. So this is the list of our collaboration, both within our institute and in other institutes outside of the campus, and also outside not only Europe but also in Taiwan or other. So if you are interested in glyphosines, please contact me, and we are welcome to visit our laboratory in the afternoon presentation. Thank you very much.
Thank you, Ivana. And the next presentation will be very young laboratory, which was established this year, truly this year, last year, but you got some rooms to this year. And it's the Laboratory of Pancreatic Islets Research and Blanca Hollando.
Thank you. First of all, I would like to thank Olga and the Institute for organizing this meeting, because I think it is a great opportunity for you to come here and to see what we are currently working on. the project that I'm going to talk about and what we really would like to offer you for either master or PhD thesis preferably is the crosstalk of glucose-induced lipid metabolism and redox homeostasis in beta cell physiology and pathology. And currently, our team comprises one senior researcher, one junior researcher, 2 PhD students, and a master degree student. And all of you are welcome to come in the afternoon. So, as the name of our department suggests, we are dealing with pancreas, and pancreas is really an interesting organ in our organism. It is composed of 2 different types of tissue, the exocrine tissue and the endocrine tissue. The exocrine pancreas is composed of so-called acinar cells, which are producing digestive enzymes, bicarbonate, and water that are directed to the duodenal part of the intestine. And then we have the endocrine tissue, which is localized into the so-called islets of Langerhans. And these are composed of different cell types. 80% of the islets in rodents are composed of beta cells, which produce insulin. Then we have the alpha cells, which secrete glucagon, the delta cells, which secrete somatostatin, and F cells or PP cells, which secrete pancreatic polypeptide. And all these hormones are really important for metabolism and are produced into the bloodstream from where they are directed to other tissues. So we are dealing preferentially with beta cells, and beta cells are highly differentiated cell type that serve as sensors of the blood concentration of the blood concentration. This is happening via binding of the glucose to the glucose transporters on the cell surface, from where the glucose is transported to the cell, and there it is converted through glycolysis to other metabolites, which provide substrates for the TCA cycle and oxidative phosphorylation. And of course, I think you all know that as a result, there is the increase in the ATP to ADP ratio in the cells. And this is the main trigger which starts the insulin granule exocytosis from the cells. Except for the ATP, there are also other important compounds that are created during the metabolism in the cells, and I would really like to mention the reactive oxygen species, which our lab in the past uncovered as one of the also main triggers of the insulin release, and these are really necessary for the insulin release. So it's not just about that they are causing harm to the cells, but they are also important signaling molecules, and also the lipid compounds. However, because our sedentary lifestyle and Western style diet that is rich in saccharides and lipids, so the beta cells are forced to produce more and more insulin, and also because there is a huge metabolic pressure, they produce more and more reactive oxygen species that are at the final stage are not able to buffer. So the ROS are then causing damage to these cells, and also lipids are deposited in higher amounts. And these as well, all these causes disbalance in beta cell physiology, and at the final stage, they are no more able to handle the metabolic pressure, and they fail to secrete insulin, and this is how the type 2 diabetes is established. And of course, it's a currently worldwide problem that like millions of people are suffering from this disease, and new patients are occurring every year. And now for the topic of the project, so how glucose and lipid metabolism is connected, and how is this important for the pancreatic beta cell physiology. So glucose and lipid metabolism is connected through the glycerol 3 phosphate, which is produced in glycolysis. And this creates the backbone for the triglyceride synthesis in the so-called GL-NEFA cycle, which is really an important metabolic cycle within the beta cells, because on one hand, it's able to store the lipids into the triglycerides, but also from triglycerides, the lipids are released, and they also serve as important signaling molecules for the insulin secretion. So the questions that we will be asking during the solution of the project is, what is the exact connection between glucose and lipid metabolism? Because this is just the partial answer I was giving you here. How the redox environment affects this connection, and what is the role of the lipid synthesis and degradation in the creation of signals for insulin secretion, and how different metabolic conditions affect lipid composition and cycling. These are three main experimental models that we use in our laboratory. The first one is the cell line INS1 E cells, which is the immortalized beta cell line, where actually all the experiments are validated and new experimental strategies are tested. When we are ready with these, so we transfer all these to the experiments on the pancreatic islets, and at the final stage, some of the hypotheses are also tested on the mouse model. In our case, we are using the Black 6J mice. And for the methodological approaches, we have a lot of different methodologies established at our department, and these in particular will be used during the project solution. So it's the handling with the RNA, involving RNA isolation, qPCR, and transcriptomics, then protein handling, like protein isolation, electrophoresis under denaturated conditions and Western blotting, also ELISA assays, and the different omics studies, including proteomics, metabolomics, and lipidomics. And at the final stage, also the functional analysis of the pancreatic islets by semi-automated perfusion assay, where we are able to measure the release of the insulin in time. And with this, I would like to thank you for your attention, and I will be more than happy to welcome you in the afternoon in our lab and to answer all your questions about the project. So thank you.
Thank you, Branka. And the next presentation will be Laboratory of Bioenergetics, given by Alena Pecinova.
Okay, so good morning. I would like to introduce you to a Laboratory of Bioenergetics. As you know, as you can see, the head of the laboratory is Tomáš Mráček, and together with me, also Petr and Lukáš, we are like senior researchers in the lab. From this picture, you can see that we like students. Unfortunately, some of them are just defended their thesis, or they plan to do so. So we are searching for the students, whatever level, bachelor, master, PhD, but we prefer to discuss with you in person the topic. So the best way would be if you come in the afternoon. I will show you the topics we are interested in. So if you are, if you would like to work on this, we really would be happy to welcome you in our lab after lunch. So what's bioenergetics? It's a field of cell biology or biochemistry, which concerns the energy flow or transduction in living organisms. And in the center of this process lie mitochondria. I hope that you know that mitochondria are cellular organelles that have a number of important functions, including aerobic respiration and energy production in the form of adenosine triphosphate. They are also involved in cell differentiation and cellular death. You can imagine that this function of mitochondria may affect especially tissues with high energetic demands. They are involved in rare inherited diseases. And as you heard, or as you can see in the previous talk, as a metabolic hubs, they are included or they associate with diseases like type 2 diabetes or metabolic syndrome in general. Our research focuses on so-called oxidative phosphorylation system, or OXPHOS. You can see it here. The first oxidative part is respiratory chain that oxidizes different cofactors that are produced by metabolic pathways, and it provides energy for ATP synthase that phosphorylates ADP and thus provide the energy to the cell. ATP synthase is a multi-subunit enzyme and it forms higher oligomers. It's also involved in organization it's organizing component of mitochondrial cristae. It has several parts, and it's encoded by both nuclear and mitochondrial genomes. The disorder of this enzyme leads to severe defects that affects newborns and lead to the deficiencies of the tissues with high energetic demands, as I mentioned before. Here you can see the assembly scheme, which is kind of complicated. And our lab, a couple of years ago, studied like group of ATP synthase deficient patients, and we found TMEM70 as a disease-causing gene. Later, we followed and found that this gene is involved in assembly of ATP synthase, and then we created mouse model, and we found that this model recapitulates the human phenotype, which means like altered ultrastructure of mitochondria, which you can see here. The cells are, the respiration of the cells is affected, and also ATP synthase amount is decreased of the assembled ATP synthase. But there are also other deficiencies of ATP synthase where the human and rat model or human and rodent model are not that perfect. One of these example is the mutation in DAPIT protein, which is the part of this, the ATP synthase, like structural part. In patients, you can see, there is a severe neurological disease, and it's caused most probably by the lack of ATP synthase dimers. On the other hand, when we created a model with DAPIT knockout, or where the DAPIT was lacking, we found that, interestingly, this model is resistant to metabolic syndrome. The insulin sensitivity was better, and so on. So mouse is not, or rat is not human, was our conclusion. But anyway, this model is very interesting regarding actually the insulin sensitivity and resistance and so on. So in these common diseases. Our other interest is in cytochrome C oxidase, or COX, which is the kind of pacemaker of respiratory chain that reduces oxygen to water. And as I mentioned, the energy is released during this process. The core subunits, as well as the catalytic mechanism of this enzyme, are highly conserved between prokaryotes and eukaryotes. And our main question is, why would we need additional components, which you can see here in like shaded? One example is the COX-4 subunit, which is one of the structural part of cytochrome C oxidase. And it has two isoforms, 1 and 2. And if you exchange 1 for 2, the oxygen or oxygen affinity is decreased. And this may be very important during hypoxia or oxygen sensing by carotid bodies. Okay. So here you can see that oxidative phosphorylation system is composed approximately by 83 subunits, but for its assembly and biogenesis, you need around 200 factors. And of course, mutations in genes encoding for these factors are like genetic cause of rare but severe diseases. Here is the example of surfan protein that leads to release syndrome, and it participates in cytochrome C oxidase late assembly, let's say. As I mentioned that mitochondria are metabolic hubs. The last topic which I want to present here is their role in adaptation or adaptations of cellular metabolism to oxidative OXPHOS deficiency, let's say. Here you can see, it's just easy, like example that when the cell, when you provide the cell with glucose, it's metabolized up to pyruvate that it's uptaken by mitochondria, goes through TCA cycle. The cofactors are oxidized, ATP is produced. But what happens when the OXPHOS is deficient? In the very last work or recent work, we found that in this case, the metabolism is rewired. Glucose or glycolytic activity increases and starts to be the main producer of ATP. The TCA cycle is rewired, and it leads to lipid accumulation. So this is, I think, our newest project. And regarding the methods, the portfolio of the methods we are using is quite broad. We are combining like old approaches as SDS or native electrophoresis with modern proteomic techniques like label-free quantitative proteomics or complexome profiling. We measure mitochondria and cellular functions. Here is the example of cellular respiration measured by 2 different methods. We measure glycolytic activity and so on and so on. And we combine these methods with the metabolomic approach, where we are able not only to measure steady state levels of different metabolites, but also to trace different labeled substrates, and we can see in which part of the metabolism it goes. So this is everything from me. If you are interested in any of these, please come and discuss, and I'm looking forward to see you.
Thank you. And the last presentation will be from Michaela Tencerova, head of the Laboratory of Molecular Physiology of Bonn.
Thank you, Olga, and also thank you all that you manage all this first block, and I'm the last person to keep you away from lunch, so I will try to be fast. So on this organ system that we are also focused, we are trying also, and I will also bring you a little bit detail on the project that we are now interested. Also, it's an ongoing project. It's based on the data that we also produce in a couple of years. And now we are really trying to understand more. So, yeah. That also my colleagues already mentioned that the metabolic complication, obesity, diabetes brings the problem all on a multi-organ level and affecting the function of important organs that are important for the glucose homeostasis. And the other organ that suffers is also bone. And I know if some of you know that we have even the adipose tissue in the bone marrow in our bones. And it can actually affect the quality of the bones and also the accumulation of the fat, like, you know, that it's coming even early before aging, can also associate and induce the higher risk for the fractures. And we all want to be healthy and to have, like, you know, this healthy aging to the high level. So would like to also really understand the regulation of the good bone quality. And that it's not surprised that there are some sex differences in terms of the regulation of the bone homeostasis. And also we know that it's also regulated based on the sexual hormones. So this is very important to understand how to treat also the complication related to bone homeostasis, such as also osteoporosis or osteoarthritis, that is different between male and females, because there are different also details about the homeostasis and the bone mass, that it's different between males and females, that we need to also understand. And the peak of the growth of our skeletal system reaching a different level between males and females, and also how the decline of the bone mass really after removal of the estrogen level or other steroid hormones really affects this impact, that it's really huge. That's why it's also important for a future, like, you know, in designing the really good drug therapy to prevent this decline. From our previous studies, we also would like to approach it with animal models that we have. It's what was already mentioned by my colleagues. It's usually this Black 6 mouse model with high fat diet induction to induce this metabolic complication. And also one of the complication with bone metabolism is also impairment of the structure and the quality, and also accumulation of the fat. What we also found in the studies between males and females is that you could see that both of these models are having this impairment of the glucose metabolism, but in males it's much more robust, like, you know, this glucose tolerance between a normal and high fat diet. And also the depot distribution between these two sexes, but the accumulation of the fat is also different between males and females, which also really intrigue us that this is something regulated also in the mouse models, which have still active, you know, the steroid hormones present in their system. So in females, we can actually observe with high fat diet much more accumulation of the fat, even in the normal condition. So this really got our attention, and we would like to know the molecular mechanism. So that's the also place for you, like, you know, to coming in the lab and really work on some interesting data. We also found on the cellular level, because the bone marrow adipocytes coming from the bone are actually differentiated from the bone marrow stomach cells. So when we actually isolated these cells from normal and high fat diet males and females, we found much more robust changes actually in males than on females. So we would like to really know what is happening behind these changes in the bone marrow microenvironment. So the project is really focused on going in much deeper analysis of these two models with high fat diet, that we would like to also approach with different interventional studies in these mice, and to compare males and females, how they are responding, because it's really something that really caught our attention, and also in future for the treatment of this civilization, you know, metabolic complications, that the you know, efficacy of the drugs is different, and this is important. So with the methods that we would like to approach, that was also mentioned before, we would like to also look at the bone and fat metabolism approaches with these different omics data. But to bring you a little bit closer what it means. So the bones are, you know, with hard cortex, hard to see what is inside. So we really need to use the different bioimaging and some methods that can give you an answer about the structure, what is under this hard core, which is really correlated with also the quality and the mechanic properties of the bones. So we are using the machines like DEXA scan or micro CT, and also some histological sections to really look at what is inside of these bones and to look at the images with different staining agents, look at the adiposity and vasculature inside the bones, which is really interesting and cool method. And also the mechanical properties, as I mentioned, it's to see the fragility risk in these different models. And then what I mentioned is also to look at on the cellular level, like, just how these cells between, these different models change. So we are isolating the bone marrow stroma cells from the bones of the mice. We are also having a collaboration with clinicians to get also samples from patients that we also in a different context and different metabolic status can compare their metabolic properties, their differentiation capacity, and also some other molecular analysis that is done in the lab. So with this also, I would like to invite you in the afternoon if you would like to see much more. We have now even in culture some human cells if you would like to see, and also what we are doing. So thank you for your attention.